Bonjour à toutes et à tous. Welcome to this very last webinar of the season. Of course, the SEM is organized by the LABEX CEMEB, and we thank Agrinium for helping us with setting up the webinars. So as you all know, and maybe for those who are new, for the questions, you can ask them during the seminar. If you look at the bottom of the Zoom window, you will find a tab that is called Q&A, and you can simply type your questions there already during the seminar. And you can also vote for other questions that you haven't asked so that we know what kind of question is most interesting to everyone. There's also the chat that you can use in case you encounter any technical issues, for instance, and you want to communicate with us. And with that, Alizon.
Hello, so yeah, thank you everybody for attending the last seminar, Ecology and Evolution series of the year. So this, or the academic year. So this is a follow-up to the COVID-19 seminar that we had by Mercier Soffonia, Nicolas Bien and Sylvain Gandon in March. And today we are very pleased that Samuel Alizon has agreed to speak. So Samuel is a CNRS researcher working in the theoretical and experimental evolution team at MIVEGEC in Montpellier. So the work he's going to present today is a collaboration between people in the team. And this includes researchers, an assistant professor, PhD students, a master students, and an engineer from the CNRS, the IRD, and the University of Montpellier. So here, many of the people in the team work on infectious disease modeling. And Samuel said that investigating the epidemiological aspects of COVID-19, which is what he's going to talk about today, was very much the natural thing to do next. So we're really looking forward to what you've got to hearing about this work. So thank you very much, Samuel.
Okay, well, thanks, Ali. Thanks to the team for organizing the seminars. I will try to share my screen to show you some slides. So and here we go. It's not ideal, sorry. I will share the whole screen. It will probably be better. I can't seem to be able to share the whole screen.
We see.
Yeah, it's a bit truncated.
For you, but not for us.
Are you sure?
Yeah.
I think you're going to miss some of it, and it wasn't like that before when I tried. Anyway, I'll start. So yeah, this is a seminar entitled COVID-19 Crisis Epidemiological Modeling. The crisis is both about the study object and about the conditions under which we perform this work. It is definitely a teamwork. I'm going to speak on behalf of a bunch of colleagues. Thomas Beneteau, he's a PhD student working on HPV modeling and cancer. Marc Choisy, he's heading a modeling team in Vietnam. Gonche Danesh is a PhD student in PHYLODYNAMICS. Ramses Jiju-Demas is an IRD researcher. Baptiste Elie is a master student. Believe it or not, his master project was on outbreak modeling. Yanis Mechalakis is a football player from Saint-Gély-du-Fès and also a CNRS researcher. Bastien Régnier is an engineer in the team. Quentin Richard is a postdoctoral fellow. Christian Zellinger is an IRD researcher, and Mircea Soffonia, who talked in March, is an assistant professor. And so, yeah, we're based in Montpellier. I thought I would begin with a timeline to try and put things into context. So time goes from left to right, and I've shown the total number of cases, confirmed cases at each date, and this is a log scale. As you can see, The epidemic started in China. So initially, all the cases in the world were in China. January 12th is an important date, especially for us evolutionary ecologists. It's when the virus sequence was published by CDC China. So less than 2 weeks almost after the onset of the epidemic. And this is a new virus, so it's particularly impressive. January 22 is when the WHO confirmed interhuman transmission after visiting China, and the lockdown in China followed that conclusion. Then there's a large interval during which, well, the main event is the WHO recognizing the epidemic as a public health emergency of international concern. So it's really at this time that the alarm bell was really rung. Nothing really happens. You can see there are a few cases. The first cases in Europe were detected in France from travelers. France declares that the epidemic stage 2 is reached on February 29. Epidemic stage 2 is when you have transmission chains in the country, but in theory, they are under control. On March 7, the government advocated basic measures, social distancing, not wearing a mask, for the general population at least. March 9 is another important event because it was the lockdown in Italy. So you can see the blue curve in Italy, which started to peak before France. Shortly after this, the WHO called it a pandemic. And still in France, the municipal elections were maintained on March 15th, which I didn't write. Schools were closed on March 16th. And in fact, the country went to lockdown on March 17th. And for Germany, it followed a week later. In our case, in the team, we started working really on COVID around March 12th. So right after it was a pandemic. Of course, in terms of timeline, I have to mention that on March 5th, we were demonstrating and on strike against the LPPR, a law for the research sector and higher education, which has been unanimously criticized. There's little I can add about the terrible aspects of this law. I encourage you to read the report from the CESE that was published recently. It's very, it's very sharp, and I find very accurate. I think you don't have the whole address, but you'll find it online. What I can add on a more personal level is that I'm particularly shocked by the contempt and disdain by the government. I mean, seeing a ministry for research saying things like in Libération, having a PhD never exempted anyone from taking an exam is just mind-blowing. And so in the team anyway, we decided not to answer journalists anymore when they want us to tell them about the ongoing epidemics, which we've been doing since March. And a lot of this have actually been private lectures for journalists. You really see a difference between serious journals that have a science service and journals who just discover COVID every day. Okay, back to Montpellier and the team. So initially in an epidemic, there's little you can say. This is what basically the data looked like early on. So this is the number of cases. Again, it's a log scale as in the timeline. So a linear increase is actually an exponential growth. Early on, you just have isolated cases. So this red-ish area is kind of when the epidemic is, the stochasticity is really strong. You're not even really sure you're going to have an epidemic wave. And then you switch to something that's more deterministic. Some of the things you can calculate there, it's the classical stuff, but through the slope of this line, you can estimate the exponential growth rate, which is also transformed usually into a doubling time, which is more intuitive. So the doubling time was 3 days. It means every 3 days the epidemic doubles in size. Of course, initially you go from 2 to 4 to 8, etc. But at some point you have to realize you can switch from 1 million people being infected to 2 million in 3 days. So this measure is actually more informative, I find, early on than the R0. but it really tells you how fast the epidemic was growing. The R0, I guess everybody's familiar with this now, except the Ministry for Research, who called it a probability. So given that it's greater than 1, it can't really be a probability. There's an intuitive interpretation to R0, which is the average expected number of people infected by someone during his or her infection period, provided that the whole population is susceptible and that there's no control measures. And so we estimated it early on. We wrote a report on March 14th. We estimated it around 2.5, and that was taking data between February 27 and March 16. Actually, if you restrict the data to March 9th, you find a much higher estimate. What is indicated here is just to show that this is really basic thing, because the data we took from Our World in Data, which was more or less the WHO data, we used the published R package, and we used a serial interval. So to calculate the R0, you need to know the time between 2 infections, so how many days. And this data was not available for France or Europe at the time. It still isn't, actually. The only available data still is coming from Asia. And so this was the data we used. So that was March 14th. I think it's one of the first estimates for R0 in France. The government officially, the first official announcement on R0 in France was made on April 23rd, I think. So yeah, it was quite early in the epidemic. And it's, in a way, it's kind of natural for ecologists and evolutionary biologists to be working on these issues, because it's really population dynamics at heart. And here to show the parallel, I've written the reproduction number, the temporal reproduction number. So the difference with R0 is that this is during the epidemic, and you can really write this as following Euler's equation. So in concept, the idea is that you need to know what's the probability distribution for individuals to survive until age A, what's the distribution of having offsprings at age A, and then, well, you can this way estimate the R, which is your expected number of offspring. There are some difficulties, because typically with epidemics, you work in discrete time, there's no continuous time. And also very quickly you leave this exponential growth regime. And so then, but then still you can find approximations to express this reproduction number. So here, Y of t is the incidence, so the number of new cases you detect at time t. And basically the intuition is that the reproduction number, you will be looking for what's on the denominator here. Yt minus a is the number of new cases you had a days ago. And so you're looking at whether these cases can have produced some of the case you observe at t. So you're basically summing all the cases that might have led to the case you observed at t, and therefore you know how many cases were caused by one infection on average. What we did, so it's since April 17th, the slide is cut. What we did was that we created an online application for people to visualize the variations in this reproduction number. And this is done for different French departments or regions for different countries, and you can also use different type of data. Again, this has been online since April 17th, and to my knowledge, it's one of the only apps that allows you to visualize these trends in reproduction number directly. Even at the international level, there aren't that many that do that. One of the problems with having a national value for the reproduction number is that it can mask a lot of heterogeneity among areas. And so since the end of April, we now show on our website a map of France showing the reproduction number. So basically when it's black, the epidemic is growing, and when it's dark blue, it might be growing. Light blue might be decreasing, and light is clearly decreasing. The dashed areas is where there's not enough data to even compute the reproduction number. And so this is the latest map we've put online. It's the state of France early June. And here you can see that the trends you detect in France may be strongly affected by what's happening in Guyana, where the epidemic is strongly growing. And then you also see that the east of France is still more affected than the center or the west. Okay, another type of data we were able to use early on came from virus genomes. So it's kind of amazing how, because we often tend to show the negative aspects or focus on the negative aspects, but on the positive side, the data sharing when it comes to genetic sequences was really amazing in the continuity of the Ebola epidemic. So I said the first genome was available on January 12th. Here you can see that on, this is just for France, but on March 27th on this platform GZAID, which was originally created for flu, there were 75 genomes. Overall, worldwide, I think we're above 50,000 genomes contributed now. And so having access to these genomes as well as their date of sampling is really useful because you can apply techniques that have been developed over the last 20 years in the field of phylodynamics. Indeed, by analyzing the underlying idea is that there's an intuitive link between an epidemic and a transmission chain or a transmission tree. So here times goes from top to bottom. And here we show classical prevalence. So the number of cases at a given time. These are just 3 outbreaks with the same parameters. There's just stochasticity. But on the right, we show a transmission tree that corresponds actually to the black epidemic here. And so every time you have a new case in the black epidemic, you have a branching in the tree. And every time the prevalence decreases, you have a leaf. And so you can see there's a direct link between the two. And you also see that clearly there's more information in the phylogeny, because if you know the phylogeny, you can recreate this, this prevalence curve. You just need to sum the number of active branches at a given time. But on the contrary, if you're given a prevalence curve or an incidence curve, which is usually the data you have, you can't really infer this phylogeny. So there's more information there. And what methods have shown over the last 10 years is that you can use these virus phylogenies or these infection phylogenies to do things like dating common ancestors, so dating events in the phylogeny, quantifying the spread of the epidemic, so the doubling time or even the reproduction number. You can also sometimes access infection duration, so or more precisely how long someone is infectious for. And finally, you can investigate the geographic structure or more generally the structure of the epidemic. So we did this with the French data. We published a report on March 30 based on 69 sequences from France. So these sequences were shared by the national reference centers. Not all of the people who did sequencing shared their data. I won't give out names, but anyway, so the reference centers shared their data. And you can already have an idea of what's happening in the structure of the epidemic there. Indeed, you can see there's, so time goes from left to right, except here the phylogeny is not shown, is not rooted in time. It's just a number of substitutions. But typically you see that the early events, the early sequences seem to be on the side, and you have a central clade which contains most of the recent sequences. And so this kind of suggests that there were initial introductions of the virus in France. Here you have the very first cases that were detected in January, but these typically didn't seem to have contributed to the epidemic wave. The epidemic wave is here. I mean, what we think is the epidemic wave. You can see that you find several French regions. The colors corresponds to the region. I'll come back to that later. And well, you can see that clearly there have been multiple introductions of the virus in France, and not all of them have contributed to the epidemic wave. Here we dated the origin of this clade between January 18th and February 13th, but it's there's a lot of uncertainty, and I'll come back to that. We also estimated the doubling time of the epidemic, of the main clade here, and found 2.5 days, which is kind of consistent with a number I gave you from the incidence data that was 3 days. So here it's just using the genetic data, you can infer that. Almost, I think the day we published our report on March 30, lots of sequences became available. And so we redid the analysis, but this time with 196 sequences. 86 of these were in the main clade. So we could refine a little bit the origin of this main clade. Note that there are limits to the origin of this epidemic wave. First, as you can see, not all French regions are represented. Occitanie is not in there. So perhaps if you would add sequences from Occitanie to this main clade, you might need to go backwards in time to find the common ancestor. So that would bring your date of onset of the epidemic wave a bit early, although not very early, I guess. And on the contrary, here you see that we have a polytomy here, where we're not really able to discriminate what happened. It could be there were multiple introductions of the virus in France. It could be an initial super spreading. What is clear is that if there are multiple introduction events, sorry, it means that the common ancestor we're finding here is not necessarily the first infection in France, but rather the infection that generated the infections that seeded the wave in France. So you have to be careful about these estimates. As I was saying, you can go a bit further with more sequences, and using the BD-Sky model developed by Tanja Stadler and her team via BEAST, we were, for instance, able to measure the reproduction number during 3 time periods. So the first time period, the prior is the dashed line, and you see that the first reproduction number you estimate is not very different from the prior, so you can't really say anything. This is for what's happening in February. Then we have in yellow we have what's happening between March 7 and between mid-February and March 7. And so you can see that here the reproduction number we infer is very high. The median is around 2.5, which again is consistent with what we get from the incidence data. And finally, the third period, which is from March 7 to March 24th, which kind of includes part of the lockdown in France, you find a smaller reproduction number closer to 1. So it's very consistent with the incidence data. And you can also try to infer the duration of the infectious period. In gray, this is the prior distribution we have, and in black, this is the posterior distribution. And you see that the median is around 5 days. And this is very consistent with other estimates for the serial interval, for instance. So it's an illustration of what you can do with sequence data. Unfortunately, since the end of March, the number of sequences in France has really become limited. Here, France is in red. And you can see that it was growing, and then we're really at a plateau since May. The UK, on the contrary, has been flooding the database. This is the GENOMES database, has been flooding the database with genomes. And this is the percentage of the genomes from Europe that were contributed from France. So you see that early on, France was contributing most of the sequences, which is obvious since this is where the epidemic started. Well, compared to the UK, but then it dropped, and now we went from 40 to 10, and now we're around 1% of the sequences. Another unusual fact about this epidemic is that, well, in a way, the crisis kind of exacerbates all the weaknesses of the health system, but also of the scientific system. And so here, one of the things you can visualize is just the number of papers that have been published or put on preprints since the epidemic, with a peak. This is per week. You can see that the week of the 20th of May, there were more than 2,000 articles published. So it's really it's really major publication event, and it raises lots of issues. For instance, one of them is the fact that nowadays you have to pay to publish in most journals with these gold open access. It raises questions about peer reviewing, because a lot of the COVID papers are published with a rapid peer review, and it raises all the classical concerns about plagiarism or fraud or whatever, are again exacerbated in this context. Anyway, because I was mentioning this because the analyses I've shown before are more public health analyses, and not really material for publications, I would say. It's more, we just applied existing methods to the data. However, we did do some more original modeling. So this started during our first meeting on March 12th, and we created this this model, which in the end turns out to be not that bad. Where this is that's what we do in modeling. You know, it's the master student internship. That's usually one of the first problems we give them, is writing the life cycle. So here you see that susceptible host, we assume that upon infection, they will either develop a light infection, or so a mild infection, or a severe infection. And then they're first exposed, which means they're infected but not infectious. Then they move to an asymptomatic phase where they are infectious but not symptomatic. And finally, a full-blown infection where they're both symptomatic and infectious before recovering. And it's the same for the severe infection, except with the severe infection, there's a probability to die. With these models, you can do lots of things in terms of conceptual modeling. You can, for instance, write R0, which I presented before. The interesting thing is that now your R0, you express it as a function of the various parameters of the life cycle. So you can see which parameters are important, which stages of the life cycle of the virus contribute to its spread. Going a bit further, what we could do, this is work mainly from Ramses Demas, was to apply optimal control theory. So one of the striking things about the COVID is that there wasn't, and to a large extent still isn't, any treatment or vaccine, which means that the only way to control the epidemic is through non-pharmaceutical interventions. And this is where you need modelers. And so here what we try to do is to see, well, assuming that you can control the spread of the epidemic, whatever this type of control, this is parameter C. So C is the intensity with which you decrease the transmission. If C is 100%, then you completely suppress the transmission. So assuming that you can control the epidemic, how should you exert this control over time? So you have a fixed time window. We take approximately 2 years, saying you have 2 years until you find a vaccine that works. During 2 years, all you can do is have these control interventions, whatever they are, that decrease transmission. How should you implement them? So mathematically, with optimal control, you're looking for a function C of t that, so notice that it's not just one parameter. You're not looking for, I want 10% of control, I want to suppress 10% of transmission, but rather I want to control with that amount of effort at that given time. So your intensity of control can vary through time. And what you want to do is minimize this objective function G here, which, without going into details, has two components. The first one captures the number of deaths, either directly linked to COVID or indirectly, because here in the model, we assume that if the number of people in ICU is above the threshold that the system can sustain, you get additional deaths. So anyway, so these are the deaths on the first term. And then on the second term, it's a term that captures the cost of implementing the control, especially with this parameter B. So the smaller B is, the easier it is to impose a control on your population. On the contrary, if B is high, it means that the population will really not appreciate being under lockdown or whatever. And then it means that every unit of control is more costly. And so using optimal control theory and Pontryagin's principle, you can find an analytical solution, well, with numerical integration, but you can find the optimal solution. And so this is what it looks like. So on the top panel, this is the intensity of the control during 100 weeks. And this is a more classical representation of the prevalence of the epidemic during these 100 weeks. In black, it's the situation where you do nothing. And so if you do nothing, the epidemic will grow. You will quickly be above the ICU bed number capacity within 20 weeks. And so you get like, it's the worst case scenario with a peak after 40 weeks. Now you can try and implement control. And so in blue, in light blue, it's the optimal control. So this is what is shown here. You see that this optimal control, it's a bit hidden. I'll explain after what the other curves are. but you basically increase really rapidly your control, and then you increase steadily before relaxing the control towards the end of the control period. And with this, you see that you completely suppress the epidemic peak. This is the blue line. You remain below the threshold. The gray and blue line are discrete implementation of this optimal control, because here this is a continuous solution, but in reality you can't change the control level every day or so. It's unfeasible. So here we looked at whether we can approximate the optimal control, but with updating the strategy only every week or every 2 weeks. And you, as Ramses found out, you see that it works quite well. And just to compare, you can compare what would happen if you would just use a constant control. So after a delay, you just control the epidemic with a constant effort. And we chose this constant value such that the area under the curve is the same. And you see that the constant control works more or less well, but at some point the epidemic grows too much and you're above the threshold. And so here we're just representing the number of deaths. These are direct COVID deaths. So you can see that if you don't control, there are lots of deaths. And otherwise, these are the indirect deaths. So due to hospital saturation. And again, you see the constant control, which eventually leads to some deaths. These are some more investigations showing that if you control earlier on, you can have different patterns, but I'll skip this and move to the second main point I wanted to mention, which is that this simple model we published, this toy model, it has a big issue, which is that it's memoryless. It's the classical term is Markovian. It means that if, for instance, you've been infected for 1 hour, you've just moved to this E2 class, you're as likely to move to the A2 class as someone who has been exposed for 2 days or 3 days. So there's just no memory in the process. These are exponentially distributed waiting times. And it's kind of an issue because we know, of course, that you won't show symptoms 1 hour after being infected. And similarly, you won't remain without symptoms. It won't take 2 weeks for you to show symptoms. So these delays are really important. And this is what we show. This is work from Mircea Sofonea. Here, what he tried to do is to fit the data using a Markovian model. So using this model, he took the daily number of ICU admissions, these are the blue triangles, and the daily reported deaths. So you see that there's a delay between the two because you first show symptoms and are admitted to ICU before dying. And this is as good as it gets with the fit, with a Markovian model. Basically, you can't fit both. Of course, if you're only trying to fit the deaths, you'll do reasonably well, even though I'm not sure you'll capture the steep decrease. And if you want to fit the both time series, it's just undoable. And the reason for this is because you don't have these memory effects. And so what we did was to introduce that in the model developed by Mircea, so that essentially the structure is the same as the one I've shown before. You have susceptibles, you have mild infections, you have severe infections, you have the recovered and the deaths. But now you see that it's a discrete time. So when you get a mild infection, I refers to the age. Well, first you're infected at day 1, then you move to day 2, then to day 3, etc., then to day G and J, and before eventually recovering. Same thing for the severe infections, and then severe infections always lead to hospitalization. Then we have 2 trajectories, which I can come back to, but basically there's one where you're guaranteed to die, and another one where there's just a probability of dying. But the important thing here is that in this discrete time, there's a history. Your infectiousness varies depending on the days, and you will have to move from one day to the other. You won't directly recover from the infection in one day. This model, or an earlier version of the model, was implemented online. This was on April 6th. Sorry, the slide is cut. Here I'm showing the new version that should be online soon. which implements some additional details. And in the new version, one of the things we found was important was to allow the user to vary the control strategies, to see, to basically get an idea of what the options were in terms of control, and they were kind of limited. And here we've implemented some additional things, so you can have a massive screening or not, with the test and screen being able to reduce the transmission. And you can also implement some adaptive control with stop and go, and you can have different age groups, different control over the age groups. So it should be online soon. And so about this parameter estimation, so the way we've proceeded, like most of you, before March 27th, we were just, Mircea was watching TV and writing down what was the new number of infections and building our little spreadsheets. On March 27th, there was a change because the incidence, so the number of new cases hospitalized or in ICU, were published online through an official website. April 2nd, they published the official number of deaths in the EPAD, in the senior residency houses. And on April 23rd, that was a big switch, because they also eventually published not the raw data, but at least gave some insight about the time between infection and potential admission to ICU, and the time spent in ICU, as well as the age of the patients in ICU. We had asked for this data in early April, but we're just a random modeling team. So yeah, on April 23rd, we were able to finally properly parameterize the model. Of course, this data was already analyzed by other team, at least one other team since March. So with the correct data, it still represented a big statistical challenge. And if you're interested in statistics, I really encourage you to go through the appendix of Mircea's paper, because estimating parameters with a discrete time model is really not that simple. Eventually, we estimated an R of 3 with a confidence interval between 2.6 and 3.4. We also estimated a date of origin of the epidemic wave, which other models didn't really do. We find it around mid-January. And we also estimated an effect of the lockdown. So the estimate, it's 75% decrease. So the reproduction number was 0.7, which was eventually what the government announced. At first they had announced 0.5, but then they were more around 0.7. We did some extra analysis with the model. I'll perhaps go quickly on this in the interest of time, but what you can do is look at what happens with a potential exit of the lockdown. So if the epidemic starts again, so the figure is a bit rich, but here it's the date on which you release the lockdown, and here is the peak number. And so you can see that, then we also vary you release the lockdown on May 11, and here is the date on which you re-implement a second lockdown. So it's to see how important the reactivity is to a second wave. And the different colors show the speed of this second wave. The red means that the wave is really fast. So you see that you get higher incidences. And the green is that the wave is slower, so you get lower incidences. So here you see that the sooner you react to this second wave, the smaller the peak. And here is the date of the peak. So you see that if you don't do anything and there's a rapid wave, you will get a peak 70 days after. However, if the growth is slower, the peak is later if the growth is slower. The main message from this slide is that if the growth is fast, so in the red case, you have to act early if you want to prevent casualties in ICUs. So here it's within a month, because it's June 15th and the lockdown was lifted in mid-May. So you see that you have a month to to get it under control. Otherwise, you're at the maximum number of cases. And on the contrary, if the growth is slow, you have more time. I'll skip on this, but basically, if you try to implement age-based controls, it doesn't really work unless you completely suppress the epidemic, because as soon as the epidemic wave goes, even the ages you protect, unless you're able to completely remove them from the system, will be affected. So I'll skip on this and perhaps mention a little bit the stop and go, so the adaptive lockdown. So the idea is that if there's in countries where there is an important wave, and this is what is seen currently in the US, for instance, or Saudi Arabia, there might not be any other options than going back to a strict control, so lockdown or any other strict type of intervention. And so then the question is, how do you decide when to start this control and when to lift it? And a strategy popularized by a report from Neil Ferguson and his team at Imperial College suggested that you could have some adaptive lockdown, which is that when the number of admissions, ICU admissions are above a threshold, you lock down, and when they go below this threshold, you go back to normal. So you end up, here in black, it's the intensity of control. Here in blue, the number of ICU admissions, and here the number of deaths. Sorry, so here you completely alleviate the lockdown, so the reproduction number increases. Sorry, it's the reproduction number. So here everything goes well, you lift the lockdown, and you see that the epidemic is going to start again. And so you re-implement your lockdown here as soon as your ICU admission goes above a threshold, and you maintain this lockdown until the ICU admissions have gone there. Then you lift it again, the epidemic starts growing, and so on and so forth. And so Mircea investigated what the optimal lockdown would be. And so there are two parameters you can vary. There's this threshold in ICU admissions to decide on the lockdown that you can vary. So 5, you go to lockdown really easily. 50 means you wait longer. And here it's how much the control is relaxed in absence of lockdown. So in green, you relax everything. And in dark red, you still maintain some control even if you're not in lockdown. What you see with the triangles is that if you take 50 people admitted in ICU as your threshold, you will get a huge cumulative mortality. These are the triangles. So you need to be in this order of magnitude, say 5, 10, or 15 to switch on and off for your lockdown for it to work. And then the other message you see is that if you completely relax the control outside of the lockdown in green, then the proportion of time you spend in this relaxed control is limited. It's 20% of the time. On the contrary, if you maintain some measures like wearing a mask or social distancing during the non-lockdown phases, you can spend much more time outside a lockdown environment. In the interest of time, I'll skip on this, but many people have been commenting that they had the intuition that the pandemic must have been there in France already in December and November and in 2017, I don't know. This is just to, the basic math that if you have a doubling time of 3 days, it's the classic chessboard problem. You double every 3 days, and at some point it can't physically hold on the chessboard. And here in France, it's very difficult, assuming the doubling time didn't change, of course, to envisage a very early origin of the epidemic. So we did some stochastic modeling on this, and you can have a look at the report on that. We did the same for the end of the epidemic, because the time for an epidemic to actually end might be long, especially if there's stochasticity. Again, I'll skip on this. Just to mention some ongoing topics. We've recently, Ramses and Quentin recently finished a manuscript on optimal control, but with accounting for the age structure of the population. We've been investigating the cofactors and how they can, comorbidities, and how they can explain the differences between sex that are observed in terms of virulence. And there's a recent report written by Yannis Mikalakis on this we published recently. We're trying to investigate spatial structure, but the GAFAs, Facebook especially, although they give you access to some of their data, the data they give access to is extremely limited, so it's unclear what we'll be able to do with this. We would like to do more phylodynamics if we can get access to more French data, and obviously to do some evolutionary epidemiology, which is our, well, the topic we like most. So in summary, we've provided some general information to the general public, mostly since March 14th. We've performed what we think is the sole phylodynamics analysis on the whole of the French sequence data. We did some conceptual modeling, especially applying it to optimal control theory with Ramses's work. And finally, developed an accurate mechanistic model with an explicit memory aspect. We developed it in a very short time without access to data, and thanks to the work from Mircea Sofonea. Some very final random thoughts, but one of the interesting things of this crisis, which I already said, is that it has put mathematical modeling and especially population dynamics in the spotlight, because there was no treatment or vaccine. It also showed that there is still a lack of epidemiological modeling expertise in French public health. Not to mention the private sector, we could come back to this, but a lot of these expertise have been subcontracted to the private sector, who have little expertise on that. One of the aspects we could also come back on is how scientific research has been and is still being bullied during this context by politicians, but also clinicians, editors, and even scientists. This crisis has also shown a problem which is perhaps more acute in France than in other countries, but about the gray zone between what is a public expertise, or an expertise, I mean, for the state, and what is scientific publications. So typically, who should have access to what data and to do what. An example, for instance, is if you work with organisms like MSF, Médecins Sans Frontières, Doctors Without Borders. Very often, you know that if you collaborate with them and have access to their data, there won't necessarily be a paper out of it, because the goal is really to provide expertise and to help. So here it's something that is still unclear the distinction between the two is still unclear in France. And finally, we think ecologists and evolutionary biologists have a lot to bring, not just to discuss the evolutionary emergence or evolutionary trajectories of the epidemic, but also to understand the spread of the epidemic and in the public debate. Okay, I'll finish with the acknowledgements again to the team. Again, this is really a teamwork. I'm just talking here on behalf of the others. Thanks to the people who are, I think, underappreciated, who are all the public health workers who generate the data that eventually gets published, even if there's sometimes a delay in publishing. They collect it daily. All the laboratories that contributed the sequences, the virus sequences that shared the gem to GESAID, which allowed the phylodynamics analysis. The team from the South Green IRD cluster in Montpellier, which helps both to run the calculations and to work on the everyday basis with the GitLab, and also to host the online applications. And the region Occitanie, who decided to support us quite rapidly and generously for the future work. And with this, I'm happy to take questions.
Thank you very much, Samuel, for this very interesting talk that gives us a lot of food for thoughts. So now we will move to the questions. I remind everyone that you can type your questions in the Q&A window. Right, so we already have a few questions. I will take them in order of whether people voted for them or not. There's one by Anne. Try to open the microphone. There we go. And you can ask your question.
Even turn on your camera if you want to make it more interactive.
Okay, I'm trying. Can you hear me?
Yes.
Yes, we can.
Yes.
Okay, I don't think I can turn on the camera for some reason. Sorry, I didn't realize I was going to have to ask my question directly. Thank you very much, Samuel. It's a very, very interesting talk. And it's not my field at all, but this week you sent a link to the preprint by Quentin Richard et al. On bioRxiv about the advantages that could be, like, so the sex differences and the age structure and how measures that account for the sex and age differences could be more effective. And I had a feeling when you talked about this during your talk that you said it won't make any difference to target age classes. So I wanted to understand how this fits with this preprint that just came out.
Okay, so there's two things. The absence of difference was to suppress the epidemic. So if the goal is really to avoid a second wave and to really get rid of the epidemic, unless there's a very specific contact structure, because initially what we thought is that by only controlling or protecting the people the most at risk, we would be able to avoid the mortality. And so what the model showed is that if there's an epidemic wave, basically, even if you put more control, so by, again, control just means protecting. If you protect people more at risk, you won't avoid them being infected and you won't avoid low of mortality. The preprint is different in that here you're looking at you're looking at the intensity of the wave itself, and potentially you also allow yourself to completely suppress the wave. And so what you find there is that for a given time period, which is also a difference with the other, because the other you run it for a very long time period, the aiming for some ages might make a difference within this window. So it is a different setting. There's anyway a general difficulty for the age-based control in the practical implementation. I mean, that was seen in France, where the National Academy of Science said that you shouldn't do that. So the aiming the age has no effect if you want to completely suppress the spread. However, if you want to control it and to look at variations in the number of deaths, it will make a difference, but not a massive difference if there's a second wave.
Okay, I think we can move to the next question if Anne is happy with the answer. So, like Simon was asking, Simon, do you want to turn on your mic?
Here you go. Does it work?
Yeah.
Right.
Hi, Samuel. Thank you very much for the very interesting talk.
So it's about your question about, you know which one, right? Get ranked.
I don't know which one, but I have two questions.
The one's about contact with politicians.
Is that this one? All right. Yeah. So Samuel, were you in contact with politicians or policymakers during all this time? And apart from the journalists, I mean, and what do you think was their understanding of on the one side, the situation, and the other side, what science was and what science contribution could be?
Yeah, so I think the general answer is no. I mean, it's something we didn't really discuss, but I mean, clearly we could have from March 12, have asked to be official expert, etc. And we didn't really go into that direction. Initially, we really went more to do pedagogical notes and just explain how basic epidemiology works. And it's in the process that we decided to go for more accurate models. So no, we haven't really been in these decision centers, but yeah, it's difficult to know. I mean, the only one who has been really in the forefront for modeling is Simon Cochemez, because he's part of the scientific council, and it's difficult to know I mean, in general, it's difficult to know what the weight of everyone was. I personally was, however, involved with a policymaker because I did a seminar with Cédric Villani, and he understands modeling really well. But yeah, no, it's they actually wrote a report for the science committee at the Assemblée Nationale, which is really good, but it came quite late in the epidemic also.
Okay, so Lunes had 2 questions. So Lunes, perhaps you can choose one first and then later we ask another one, or do you want me to ask it? Lunes Chiki?
Hello.
Okay,
Hello.
il est là.
Okay,
Yeah.
hi, thanks. Well, first of all, great talk. It was really clear and it's a very nice work. So thank you very much, Samuel. The 2 questions, so one of them is actually similar to Simon Felou's first question. It's about, at some point you were showing periods of lockdown and periods of opening. And clearly, I would expect, as in graph, that the closing or the lockdown would be longer than the opening because of the exponential growth, and it doesn't decrease exponentially. But I was wondering, is there a way to mitigate that and have the more balanced possible? Because people might accept to alternate, but if they always feel that it's 1 against 10, they may not like it at all. And second was about population structure. How does that influence? You said you're working on spatial structure, but can you say a few words? Thanks, sorry.
Yeah. So for the first one, I'm not sure I understood, but you're completely right in that usually you stay in lockdown longer than you stay outside of lockdown. And one of the ways to mitigate this, so to spend more time outside the lockdown, is basically to maintain some control even outside the lockdown phase. Then we also tried, this age thing a little bit, try to see whether you could alternate between the ages or things like this. So have people who are above a certain age be on lockdown and people younger not be on lockdown, and alternate like that. But it didn't seem to have a huge effect. Although, and coming back also to Anne Charmentier's point, the difference is that in Mircea's model, there's not a contact matrix per se between ages, whereas in the other model, we have an explicit contact matrix, which could explain the difference. And which brings me to the structure question. Initially, in the early stages of an epidemic, there's some paper on this that I think Mircea cites in his paper. The structure won't really matter in a way, because the saturation doesn't play a lot. You have all these transmission chains that are spreading, and they're not conflicting with each other. Right now, it's estimated that 5%, roughly 5%, probably less, of the French population has been infected. So initially, you can just work without this structure because you don't have these local saturation effects. As soon as you reach higher prevalences, yes, totally, then it's kind of the herd immunity playing in. Once a local place, lots of people have been infected, it will spread more slowly. So yeah, for the further stages of the epidemic, you would need this. And we've been trying to look at initially more the dates of onsets in the different departments and see whether we could capture this with so Christian Zellinger worked on this, but it was it's difficult also because of the data, because you know the reported cases and there's not the same intensity of sampling in every department. So
Thanks.
All right, so now, let's see, we have several questions. Some of them don't seem to be directly related to this talk. Perhaps we'll take the question by Véronique Viader first. Véronique is here, or I can ask it. Turn on your microphone. No? Yes?
You should
okay.
be okay. I asked 2 questions. I didn't understand the link between phylogeny and all you say there are more information in phylogeny than in other numbers. So could you explain, and could we access to the publications you give at the end of the slide?
Yeah, totally. So I forgot to mention it, but all or most of what I presented is in the reports, and especially for the phylogeny one, we've also written I have a book chapter I should put on HAL that will come out soonish. There are two things. One of the things is that the phylogeny and the parallel I've shown with the transmission chain is more to get an intuition, but behind it, there's in the models we've used, it's working directly from the sequences. So it's more it's more a statistical interpretation, but still the phylogeny kind of gives you an idea. If you have a perfect sampling, so you know everyone, typically if you take the distance from a branch to a leaf, one of these corresponds to someone who was infected and someone who recovered. So the length of this branch tells you for how long this person exactly was infected. And so if you've sampled your whole population, assume there's only 100 infections in France, and you've sampled all these 100 infections, just by measuring these branch lengths, these terminal branches, you could get access to the time between the infection and the recovery, or the end of the infection for these people. This is something you don't get with incidence data. Incidence data, you just know how many people were detected today. You don't know how long they've been infected for, and you don't know how long they will remain infected for. So yeah, it's really this time component, the fact that a phylogeny is rooted in time, that gives you extra information about than the incidence. But yeah, it's difficult to answer in. But yeah.
All right, so we have several other questions, but I'd like to. So some of them are from people who had already talked. So there's Thierry Valero who asked 2 questions since the start. Actually, he had written them in the chat screen first. So I don't know if we can take the 2 of the questions. You can read them also.
I read
Thierry is here.
quickly, but
some of them are related to the study in Imperial College, but I don't know that if you have something to say about this. And the other one has more about this, I guess, the social component of the epidemics, if I understand well. So Thierry can correct or react.
Oui, en français, je traduirai en anglais.
Pas de problème, ouais.
La première question me semble plutôt fondamentale. C'est la question, effectivement, des problèmes d'interface entre la science, la santé publique et la politique. Donc, on a un modèle comme l'ICL, dont j'avais entendu parler, comme tout le monde, depuis de nombreuses années. Je suppose qu'il y a des publications scientifiques. Il a servi plusieurs fois la décision politique. Et à l'occasion du Covid, on découvre ça, la très mauvaise qualité du code au niveau du code source comme de l'architecture. Pour le dire simplement, si on a ce logiciel dans un avion, on a 300 morts au décollage. Il a déjà servi pour des prises de décision politiques qui ont touché plusieurs millions de personnes. Je pense qu'il y a un problème de fond, donc je ne critique absolument pas.
Du coup, juste par contre, le lien avec le travail qui est présenté dans leur équipe, or the link with the... Ouais.
Alors,
I mean, it's well, obviously it's not my work. So on our side, we've published all our code, and yeah, clearly things are changing. One thing is that when it comes to public decision, basically I mentioned the doubling time, which is 3 days. So you have to reuse code that you already have if you want to give something quickly, because you can make mistakes. So here it's code from a Nature paper from 2004, I think, or 2005, which also explain. I mean, I wouldn't like people to look at my code from 2003 or 2004. So it doesn't excuse everything, but it kind of explains that's why they reused it. I agree with you that it would be better to have models that are already implemented and transparent, ready to use in case there is a pandemic. These models weren't there in the UK, so they used this one. I think qualitatively, most models, this is also the difference with a plane taking off and a plane landing. This is more a qualitative result. It's not, the purpose was not to say like for instance, the one from Mircea I presented, which here you really expect this kind of model to tell you how many people will be in ICU next week. Here the Imperial College was more to get a sense of the time of spread and that sort of things. So qualitatively, I think most models that were developed later on agreed with that. But yeah, I think that it should, well, it does show that you should have models ready to be used. And I mean, in France, there just was no such model. So in France, the very first models were developed, I think, by Simon Cauchemez for really targeted for the epidemic. So it wasn't ideal. It wasn't the best model, but it was still way better than what we had in France at the time. About the second question for the social component, it's, well, for most questions, I mean, the mass of unknowns is of course greater than what we know. There's been lots of discussions about the effect of pollution, so there's so many cofactors, it's difficult to tease them out. What seems to be really strong is really the density of human populations. So human density is really one of the factors, and that explains the spread a lot. And so, of course, human densities correlate with a lot of other factors. So yeah, if you live in a property with 200 square meters and a garden and a swimming pool, you're less concentrated than if you live in an HLM. So yeah, it's difficult to tease apart all these correlations.
I guess there have been some studies showing that there's some of the highest incidence in Seine-Saint-Denis, for instance, where people were had more necessary jobs and things like this. I think there were several questions more. I mean, Simon receives the prize for the highest number of questions, so he can ask another one, which he can pick up from.
The one about stop and go was moved by mistake to the result, so you can ask it again, Simon.
Sure, does that work? I should say that I voted for my own question so that they would go up.
That's a problem in the system. Now we can.
There's only 2 people who actually voted for this question, but for me. It's the same question I asked twice, actually, Samuel. It is rather, do you think those models that you did are fairly quantitative? And clearly for the stop and go, I mean, the best case scenario is something like 50% people of the time, 50% of the time people are locked down, which is not feasible in terms of, I mean, economy and for the human beings. So it's very much how reliable do you think these quantitative estimates are, yours and those of your colleagues?
Yeah. So some of the models we developed are qualitative. For instance, the optimal control is more on the qualitative side. Others are more quantitative, and yes, the Mircea's model is much more rooted in I mean, there's a detailed parameter estimation. About the stop and go, however, I think it's still a result to be interpreted in a qualitative way. And the reason for this is that well, there are 2 reasons. First, the control is we don't have the level of detail to distinguish between different types of control. And so here, basically, we just have a very strong control, which we interpret as a lockdown, and a released control, which we interpret as a normal normal life. But potentially, for instance, you could reach a strong control without the lockdown. And the second thing to add is that one of the unknowns, we were able to see what the effect of the lockdown was. So the 75% decrease in R0. What we still don't really know is how the individual interventions, wearing a mask, keeping your distance, testing and testing people, tracking, self-quarantine, these sort of things, closing schools, how these individually act on the reproduction number, not to add potential effects of indirectly the season. So it can either be humidity, but it can also be the fact that you live more outside in the summer. All this to say that what the very strong level of controls we put in could potentially be achieved with things that are less strong than a lockdown. And similarly, the achieving a reasonable level of control in absence of the strong control, so in the relaxed period, could also be potentially achieved with less with less difficulty than what we thought.
All right, so I think with this we have to wrap up because we're on a tight schedule for lunch, I guess. So let's thank Samuel again and all the participants. Just finish because this is also the end of the season. We have an open call for new seminars at the LabX website. Or I sent an email where you can have a form and you can apply for some seminars. So these will probably be webinars in autumn, but let's hope not, perhaps not at one point. So I don't know, Lauren, you had something else to add?
No, I think that's all. Hopefully.
All right, so,
Yeah.
bye-bye.